Right, thank you very much, Fraser, and thank you to Rigaku as well for inviting me to give this webinar on FlowXl and the new facility that we've been building at Leeds. So I just want to go through, what FlowXl is, what the facility is, give some background on flow crystallization, and also to give some background onto some of the commissioning experiments that we've been developing over the last couple of years at Leeds. So the facility is located at the School of Chemistry at the University of Leeds. So this is the university here, looking very nice in the sunshine. It's quite a rainy day today. It doesn't quite look like this, but it's the School of Chemistry is attached to the Parkinson building here, which sits in the bottom right corner of the image there. And we are in and we have a number of research themes at the School of Chemistry. Our group, Professor Fiona Meldrum's group, is around crystallization and directed assembly. So we mainly do biomineralization and inorganic crystallization. So we focus a lot on calcium carbonate crystallization and things like sodium sulfate, potassium nitrate, those sorts of systems. But we collaborate strongly with a lot of the science and engineering schools across campus. So we do a lot of different types of crystallization. So we look at pharmaceuticals, we look at agrochemicals, we look at polymer crystallization. So we have quite a wide scope of experiments that we've been doing at the FlowXl facility. In terms of chemistry, we have specialization in a number of different facilities, HPLC, mass spec, and NMR. We've got a very good single crystal facility as well at Leeds. And our facility kind of fits in sort of in-situ diffraction spectroscopy. So we've got quite a strong background in synchrotron and laboratory-based in-situ diffraction spectroscopy. We collaborate quite strongly with the new Bragg Centre for Materials Research at Leeds. So if people are familiar with that, we collaborate with them quite a lot. Got a very strong electron microscopy group at Leeds as well. And also, just at the bottom here, we've got really good access to mechanical engineering. We have a very good engineering workshop. So we fabricate a lot of our own in-situ sample environments as well. So flow cells, variable humidity cells, and things of that nature. So we collaborate with a lot of people in engineering, and we have good workshop facilities and things like 3D printers and that sort of thing, 3D printing sample environments. For users. So I'll just give you some background to the project. So the actual facility was built around the idea of taking the types of experiments that you do at synchrotron environment. So things like at I-22 for a SAX wax beamline, and trying to translate that into a laboratory environment. So the idea was to create a world-first facility combining flow crystallization with in-situ powder diffraction and Raman spectroscopy. So the idea was to take, flow cells or flow devices, which you might use at a synchrotron, and translate that into a laboratory setting using laboratory diffraction and spectroscopy. The reason why researchers want to do that is because it enables us to study the structural pathway to crystallization processes. So it allows us to look at things like amorphous and liquid-like precursor phases, look at transient semi-crystalline intermediates, and how does that relate to the final crystalline products, which we would typically analyze in an ex situ manner. So the idea is to look at, in a more in situ manner, the crystallization process. So how we go from molecule to metastable phase to the final crystalline product without removing the material from the solution environment. And it gets over some of the problems of doing ex situ analysis, where removing transient species or metastable phases from solutions often results in a phase transformation. So, for example, hydrates or solvates may transform into a more stable anhydrous form simply by removing those from the solution environment. So FlowXl gives us the ability to study crystallization in a variety of environments. We have a number of traditional continuous flow environments. So we have batch crystallizers and continuous crystallizers, which we use routinely. But we also have state-of-the-art segmented flow crystallization, where we look at droplets of crystals in flow. And this is just a schematic here on the right-hand side. This is an example of a segmented flow crystallization process, where you have droplets segmented from each other in oil, which pass through an X-ray beam and allow us to collect data as a function of time, or in this case, channel position along the continuous process. And we'll look at some of these in the examples that I have today as well. Okay, so the FlowXl project was it started in April 2020, so just at the start of COVID, and it ran till April 2023. So this was kind of the phase one, which was the commissioning phase. So this was to build the device, build the laboratory, to get the systems working and running. And then we wanted to commission a number of sample environments which cover various flow crystallization applications for different researchers. So whatever people were interested in doing, we would then try and translate those crystallization environments onto our diffractometer and Raman system. And then we collected initial data on selected case study systems, and these represented a lot of different fields of crystallization. So we looked at mineralization, pharmaceutical research, agrochemical, nanoparticle synthesis, so a whole range of different systems, just to kind of give us an idea as to the sensitivity of the laboratory instrument. And in this talk, I want to introduce the facility. So I'll do a virtual tour of the facility, looking at the different sample environments. And then we'll look at two case studies. So the first one is to look at an in-situ phase transformation of an API, where we extract some of the kinetic parameters associated with the phase transformation. And that'll give us a nice introduction to in-situ diffraction in a flowing system. And then we'll look at a more difficult system, which is segmented continuous crystallization. And that'll give us a chance to look at the new frame selector tool inside of the Crystallis Pro software as well, which we've developed with Rigaku. So just some background on the actual instrumentation that we have. So we have an Xlab Synergy Custom at Leeds in a very large enclosure. We have a copper microfocus X-ray source, rotating anode source. We have a goniometer, and we have a Hypix HPC detector as well for fast data collection. So the reason why we chose this system was because it allows us to do very fast data collections, and it gives us a high flux. So typically our samples are liquids, and we have a very small sampling volume. So we use small capillary tubes, either glass or Kapton capillary tubes, and our sampling volume is very small. And so for that reason, we need a very high flux, highly focused X-ray source. And because we're looking at kinetic processes which happen relatively fast, relatively quickly, we need to be able to collect diffraction data very quickly as well. So the Hypix detector allows us to collect in a shuttleless fashion at sub-second data collection times. And again, this is the flexibility of that is the reason why we chose the system. We also have a Raman system. We have a LabRam Bariba system. It's a normal Raman microscope, but we also use a in-situ probe attachment, so fiber optic probe, and that allows us to do simultaneous diffraction and Raman spectroscopy, doing real-time monitoring of the solution concentration. So the two marry up quite nicely. We have the diffractometer, which allows us to monitor the solids content in a solution, and the Raman system allows us to monitor the solution concentration. So that marriage of the two systems allows us to build a picture of the full mass balance of the crystallization system as a function of time. So this is just an image of the actual facility. So this is the laboratory at Leeds in the School of Chemistry. So we have a normal, what we call a normal single crystal instrument, the Synergy Custom, but we have this really large custom enclosure built for the facility, and it gives us a lot of space to actually implement a lot of our reactors and syringe drivers. So it gives us a lot of flexibility in terms of the type of experiments that we can do. So traditionally in situ diffraction experiments with powders, we're limited by the amount of space that we have to be able to get reactors and different flow cells inside, close to the actual X-ray beam. The beauty of this system is that we have lots of space, not quite the same as a beamline, but we have a lot of space to be able to put equipment inside of the enclosure. So we have a lot of equipment, reactors, syringe drivers, levitators, lots of different flow cells. And you can see on the image on the right here, this is our superhead probe. So this is the Raman probe I was talking about in the last slide. So this kind of sits on an XYZ stage, and it's focused on the center of the goniometer of the diffractometer. So the X-ray beam and the Raman beam are focused at the same position. And in this case, you can see the flow cell there just in the center of the goniometer, ready for an experiment. This is another close-up of the actual superhead. So this is the Raman and the diffractometer. And this is just showing you the setup for an acoustic levitator. This is a 5 microliter droplet, which has been acoustically levitated in the center of the goniometer of the diffractometer. And we collect diffraction data and Raman data simultaneously on the same droplet. So we have people doing things like spray drying of droplets and things like that, where we track the solid phase composition of different components over time using the diffractometer. So we have lots of different types of sample environments. So in the next slide, I just want to go through the other accessory that we have with the instrument, with the diffractometer. We have a CrystalCheck S, which is, we probably use this the most out of all of our accessories. It's a really useful tool for us in terms of our flow cells. So it's useful for doing powders and protein crystals, which is what it was designed to do originally. But we use it to mount our flow cells on. So for flowing our solutions through, we basically modify an SBS plate, and we then mount our flow cells onto that and then attach that into the CrystalCheck S accessory. And that gives us a lot of flexibility around the geometry that we can collect data from. So we can adjust the theta position, we can adjust the omega angle of the sample, and adjust obviously the sample to detector distance as well. And also the fact that the collision model is built into the system allows us to do really safe data collections on our samples. So we use this for our smaller flow cells. And then we have homemade XYZ stages as well, so to translate larger systems that we have at the facility. Okay, so I'll just go through the next slides looking at some of the sample environments, so the different liquid and a couple of solids as well, sample environments that we had, but mainly liquid flow cells. So this is an example of a microfluidic cell that we have. So this is a microfluidics chip, which allows us to do segmented droplet crystallization in situ. So this has been published in Advanced Functional Materials. This was data collected at Diamond on calcium carbonate. And this was looking at crystallization in small 200 micron droplets. So you can kind of see the device on the left-hand side here. It's just a PTFE channel, which is laser cut into a channel system. And then that's sandwiched between some Kapton polyimide windows with 2 silicon gaskets. And then it's enclosed in this PMMA plate, which we then seal, and that kind of makes everything watertight, basically, and it stops the system leaking. That's connected then to syringe drivers, and we mix our reactive components together in flow to create our aqueous droplets, which then go on to crystallize downstream in the channel. And the droplets are separated by oil and by air or nitrogen, and that allows us to stop the droplets coalescing. So basically, this allows us to change the channel position, which basically changes the crystallization time, or the time of the crystallization process. So by changing the channel position, we basically change the point in time at which we're collecting diffraction data from. And this allows us then to monitor how the crystallization process is progressing. So we can access things like, qualitatively, we can access the phase of the material that's crystallizing, and if there are any transient phases which are appearing during the crystallization process. And we're able to mount this on an SBS plate as well and get that onto the Crystal Check S, and then monitor the crystallization process in situ that way. So another miniature crystallization vessel we have is this very small continuous oscillatory baffled reactor, which is very similar to the last one, except it runs in a continuous stream. So where I've been segmented, it's a continuous stream of solution. And we have these small baffles inside of the flow channel, and this gives us much better mixing of the solution. And this allows us to control the crystallization process in a more, it gives a more controlled way to optimize crystallization process conditions. So it allows us to optimize particle size distributions, for example, because the particles experience the same supersaturation throughout the entire reactor rather than being segmented. So that's why the baffles are in there. And then the channel is to again give us time information. So by probing different points along the channel allows us to probe different time points within the crystallization process. And again, this can be used for doing reactive crystallization processes for minerals, or you can also use this for doing anti-solvent crystallizations of pharmaceutical, for example, or small organic molecules, for example. So another flow cell which we use is this capillary cell. So this is a very sort of traditional flow cell which we have at the facility. This is probably our most used flow cell, and it's just a transmission borosilicate glass capillary cell, which we use in transmission mode. And we've used this a lot at the synchrotrons at Diamond and elsewhere to collect SAXWAX data for doing looking at small angle scattering and diffraction during solution mediated phase transformations and nucleation experiments as well. So just to give you a better kind of understanding of how it goes together. So it's just a steel body. The borosilicate glass capillary cell sits inside of these steel containers. The glass capillary cell is 10 micron wall thickness, and we change the diameter between 1 to 2 millimeters, depending on what the sample is, and that gives us our. We change that depending on the absorption coefficient of the material that we're studying. So for organics, we tend to go to around 2 millimeters, and for inorganic systems, we tend to do 1 millimeter path length, depending on what the absorption coefficient of the material is. And then this sort of tube holder fits inside of our steel body. That gets compressed together into some O-rings, and then we can attach tubing just through these HPLC fittings. The tubing then goes to your reactor or syringe drivers, so you can then flow your crystallization solution through the glass capillary cell, and then the X-rays impinge on the window just here. So we use this routinely on the diffractometer. This is fitted to this 3D printed plate, which fits inside of the Crystal Check S on the diffractometer, which again gives us a lot of flexibility in being able to make sure that the sample position is right in the center of the goniometer, so that our diffraction data is fully calibrated. So just to show you some of the reactors that we have. So together with the flow cells, we generally use, we have a different sets of batch reactors. We have a 0.5 litre process vessel. So that's this vessel sat in the back here of the enclosure. This is just a normal half litre process vessel, temperature controlled with overhead stirring. This version is an async product that we bought at the beginning of the facility commissioning, and we use this quite routinely, I'd say, for doing process crystallizations. So we can control the temperature inside of here. We generally do polythermal and isothermal crystallization cycling. And we hook that up to our FlowXl, so the one that I've just spoke about in the last slide. We have our glass borosilicate capillary cell here, and the flow loop just goes through the capillary cell and then back into the reactor, and that goes in a continuous loop through this pumping system. And we can also do things like connecting this to our syringe drivers, so we can do anti-solvent and seeded crystallizations in order to control nucleation and polymorphism, and then collect data as a function of time as the crystallization process is proceeding. So the other reactors we have is we've got a set of continuous continuous stirred tank reactors, which are set up in series. So these are called reactors. So we do a lot of work on this on the diffractometer as well. So these are just continuous stirred reactors. So they're 2 milliliters in volume. They have continuous stirring through magnetic stirring, and they're all linked in series as a series of reactors. And this allows us to, it gives us flexible applications to multiphasic continuous flows. So we do crystallizations of inorganics and organic systems inside of these. And you can do inline Raman spectroscopy, so you can go through the top of these through the glass windows. But we've also replaced the tubing. You're looking between the different CSTRs. We've replaced the tubing with caps on, and we do diffraction through these windows on the diffractometer. So we then get in situ Raman data from the windows and diffraction through the caps and windows, which allows us to look at the progression of the solid forms as a function of time through the different CSTRs and how that changes. So the other continuous crystallizer we have is a collaboration with the University of Nottingham with Karen Robertson's group. So we have a system called the Crake, which is a kinetically regulated automated input crystallizer. So this again is a segmented flow crystallizer. So you can see here at the bottom, this is this idea of segmenting the crystallization solution with carrier fluid. In this case, it's oil. And then separating those with air slugs or nitrogen slugs. And again, we have these caps and windows at various positions. So the solution flows around the continuous crystallizer, and at each bypass in the loop, it goes through a window, which is set up on the diffractometer. The windows are made of captain in this case, and that again allows us to probe the solid phase in situ as the crystallization process is proceeding. So again, the windows give us access to time points along the crystallization process. And this is an example of work they did at the synchrotron, where they looked at a transformation of urea and barbituric acid. We've now just built a new version of this on the system at Leeds, and we've been doing some commission experiments in the last 6 months to look at crystallization of a few organic systems on the Crake. So that's been progressing well at Leeds in the last few months. We also have a number of variable temperature and humidity environments as well. So we do also do a few powders. So we've got a homemade humidity chamber, which just has caps and windows in. We can change the humidity inside of this chamber and look at the phase transformation as a function of humidity. And we have a Lincam stage as well. So this Lincam stage again allows us to do variable temperature and humidity. And both of these fit onto the Crystal Check S as well. So we have a 3D printed mount for both of these, which allows us to do in situ work on the Crystal Check. So the Lincam stage allows us to do transmission, geometry, variable temperature, and variable humidity. So we do things like analyzing phase diagrams of organic molecules as a function of humidity, for example, and temperature. And looking at that phase transitions of those materials as well. So lastly, we also have an acoustic levitator, which we use for acoustically levitating small droplets of liquids. So we mainly use these for doing inorganic salts, or we look at things like spray dried materials. So we look at evaporating droplets and then do diffraction and Raman spectroscopy on these droplets. So I've got a reference here just for where this drawing came from. It's quite a well-known levitator, which is it's homemade. We also have a commercial one as well for sort of more controlled crystallization as well. So we have a homemade XYZ stage which allows us to translate the droplets into the camera on the diffractometer, and then we can then do crystallization monitored by diffraction and Raman for those droplets. So that's all the sample environments that we have at the facility at Leeds at the moment. So I just wanted to go through some of the case studies that we've been looking at for the last couple of years, just to give you an idea of the sort of data that we've collected, the quality of the data, and what sort of information you can extract from these types of in situ experiments. So the first example we'll look at is theophylline, which is an API. It's an inhalation medicine used to treat asthma, and it has a number of polymorphic forms. So it has 4 polymorph anhydrous polymorphs and a monohydrate form. And the monohydrate is a monoclinic channel type hydrate. And under different water activities, it undergoes a solution-mediated phase transformation back to the anhydrous form to polymorph. And the reason why this is quite important to study these sorts of solution-mediated phase transitions is because of the polymorphism aspect in pharma. The polymorphs have various solubilities, bioavailabilities, and toxicities. And so it's important to understand at which environments these sorts of material are stable or unstable, particularly during storage or processing of these types of materials. So the actual experiment we did was to take our small process vessel. So we have this little small jacketed vessel which we use with a small peristaltic pump, which we use with our FlowXl, the borosilicated glass one, which we've spoken about before. And we just basically pump our solution around our flow loop, and we analyze that using our diffractometer and Raman spectroscopy. In this case, we changed the water content of the solution. We had a mixture of IPA and water, which we saturated with theophylline, monohydrate material, which is metastable. And we did that as a function of water content of the solution. So obviously driving the water content down will increase the rate at which the material transforms to the anhydrous form. So we did that as a function of water content. We added then different mass, a 5 weight percent mass of the monohydrate batch crystals, and then we pumped that solution around the flow loop, and we collected diffraction patterns every 24 seconds with a 20-second exposure time and an 80mm sample to detect the distance on the diffractometer. So what does the data look like? So we've developed a data analysis procedure as part of this. So in general, we start with our raw 2-dimensional diffraction data images. We then do some initial data reduction. So we do spike removal, we apply mass to the data. We correct the background using a clean solvent background. And then we can do the radial integration to get our 1-dimensional diffraction patterns. We then do data processing, so we can export that data from Crystallis Pro and then do external data processing in things like Origin or MATLAB. And we can do further background removal if we don't do it inside of Crystallis Pro. We can do data visualization, so we do 3-dimensional data plotting in order to look at the dissolution and growth profiles of the material. And then we do peak fitting and kinetic model fitting to extract kinetic parameters associated with the crystallization process. So just to give you an example of what the data is like, and you know, as a, you know, to show the limits of the data collections are. This is just an example of our solution media, sorry, our calibration data for theophylline. So this is just concentration dependent diffraction data. So going from 1 to 10 weight percent. So that's mass weight percent in a slurry of material of theophylline in a water and IPA mixture. And then extracting the diffraction patterns, integrating the peaks, and then plotting those as a function of concentration. And you can see we get a very nice correlation, linear correlation for our integrated peak areas, which is the black line, and the peak heights, which is our red line here. And for this system, our limit of detection, so i.e. where we don't see any diffraction data, is around 0.8 weight percent. So below 0.8 weight percent, we can't see any diffraction above the background. So that's what our limit for this experiment is in terms of the solids. This is just some data showing our actual transformation process. So these are 2D diffraction patterns, which are corrected and then integrated to 1D profiles. And then we plot these as a function of time. So we can highlight the transformation and dissolution and growth kinetics of the two phases. And then like I said before, we do peak fitting to extract our kinetic parameters such as growth and dissolution rate constants. So the image on the right-hand side here is showing you the time-dependent data. So the red diffraction patterns at the start are from time 0, and then at the end of the experiment, when the transformation is finished, are these blue diffraction patterns here. So you can see at the start we have our monohydrate peaks, which are labeled here, which are the red diffraction patterns, and then they slowly decrease because it's metastable, and the reaction proceeds to dissolve the monohydrate material and then recrystallize the stable anhydrate form of theophylline. And if we plot those in 2 dimensions, you can see at the bottom in the bottom image, you can actually see we can extract when the dissolution process starts, which is around 25 minutes in this case. You can see there's a rapid supersaturation or decrease in solids content of the monohydrate as it dissolves, and then there's a reprecipitation of the anhydrous form, which is the stable form at this temperature and water content. So just by looking at this data, we can extract information about the dissolution and growth profiles of the 2 phases, and then we can take that step further we can start to look at the transformation profiles of these 2 materials. So on the left-hand side here, you can see the dissolution of the monohydrate form, which is the black line, and then the red line is the growth profiles, the growth kinetics of the stable phase. And so the transformation profiles indicate the start and end of the dissolution and growth. The profiles then we can then convert using these equations into alpha profiles. So these are simply just the difference between the start and the intensity values of the peaks during the transformation process. And then knowing that for the 2 different phases, we can then convert that into an alpha conversion parameter, which is this equation at the bottom. And these are just called alpha conversion profiles, and they're plotted here on the right-hand side. So these are the different alpha transformation profiles for the monohydrate to anhydrate conversion process as a function of water content. So on the left-hand side, this is the highest water content. Sorry, the lowest water content is the yellow triangles. Over to the black squares, which we have the highest water content. So increasing water content reduces the transformation rate between the 2 forms and makes the transformation process longer. So you can see that in these profiles. We can then use these profiles to then fit various solid state kinetic models to extract some kinetic parameters associated with that transformation process. So you can see here, this is just a fit of an Avrami type model to the polymorphic transformation profiles. These allow us to extract the nucleation and growth rate constant associated with this process, and also the dimensionality of nucleation and growth as well. So you can see that here for different weight fraction of water. So as we increase the amount of water, the rate constant decreases, which is which relates well to what we see in the actual experiment. And the n parameters here, this dimensionality of nucleation and growth. This tells us that we've got a three-dimensional. On average, we have a three-dimensional growth process, which is what you'd expect for this type of solution-mediated phase transformation. We have a three-dimensional growth of the nuclei and crystallites during that transformation process. So this is the type of information that you can extract from these experiments. It shows that we can extract quantitative information on the amount of phases using the diffraction data. And we can track over time the concentration of these phases using the diffraction data as well, which is really useful, other than just being able to identify the different phases present. Okay, so this is the second example that I had, which is just to show you our segmented crystallization. So now, rather than looking at a batch process, like the example in the previous one with theophylline, this is now looking at a continuous process. And here we're generating droplets or slugs of material, which we're passing in front of the X-ray beam. So this is our mixing device, which we're using to generate the droplets of material. So we have this PMMA millifluidic droplet generator. We have a carrier oil, which coats the tubing, and that prevents fouling of the tubing with the material, with the crystals. We have nitrogen in this case, which is used to control both the flow rate of the system, but also it separates the slugs to stop them coalescing inside of the tubing. And then we have these input ports, and these are all used to generate our droplets in flow. In this case, we're doing an anti-solvent crystallization. I think this case was glycine, I think. So this is using glycine, aqueous glycine, and then an anti-solvent with ethanol, I think it was. But you can change these to any of your solutions. So we do reactive crystallizations with inorganic salts as well, where we just mix two reactive components together and then create droplets that way. And then we change the time points by changing the tubing length. And so each residence time allows us to access a different time point in the crystallization process. And then we collect multiple diffraction patterns at that time point and then sum them together. So the problem with doing this is that we can't just continuously collect data like we can on the previous example, where we have a continuous stream of particles going past the beam. In this case, we have both an aqueous phase, but we also have our oil and gas phase, which doesn't contain obviously any useful information on the sample. And so we need to remove that data from our analysis procedures. So we've been working a lot with Rigaku on how to do this. And one of the main problem is kind of highlighted in this movie on the left-hand side here. So this is basically looking at raw data. So this is in real time as the detector sees it. So this is collecting data at 0.1 seconds per frame of a segmented crystallization process. And you can see that some of the frames have lots of diffraction data from them. But then other frames have no data on them, because that's the frames with the oil and the air and the nitrogen or air, whichever it is that you're using to segment the flow with. And so we want to remove those frames from our analysis procedure, and we want to only keep the frames which have diffraction on. So we have a procedure where we add those together in a hit frame. We keep all the hit frames together. We sum them to make a composite diffraction image, and then we do our radial integration on that composite diffraction image. So this actual process was originally done in this paper, which I've highlighted down here, which has the original MATLAB code on doing segmented diffraction data analysis. but we've been working a lot with Matthias in Rigaku to implement this inside of Crystallis Pro, which he's done for us, which works really well. And the general data analysis procedure is given on the bottom here, where we have a normal spike removal, mask application. We correct some parts of the images. We then do a thresholding of those 2D images, where we define a threshold value for the pixels. And we basically say if there's anything below that threshold value, remove those images from our analysis. But anything above those pixel values, we keep those and we do our analysis on our, and that they're classed as our hit frames. And then we take our hit frames and we sum them together to create our total diffraction image. We then do a background correction, and then we do an image integration to get our indexing. And this is just an example here of salicylic acid. So this is what it looks like inside of Crystallis Pro. I'll just very quickly run through what it looks like. So the frame selector tool is under the data reduction tab, and it's under the frames section here. And this brings up the frame selector GUI here. You can also access it by using XXFS, which brings up the same GUI. And this is what it looks like kind of up close. You have the frames shown here, which shows you which frames you're using and not. So the total frames here is 648 frames in the experiment. And of those 648, we're using 16 frames, which we're classifying as hits, and 632 frames, which are misses. And we've also got down here another useful feature, which is the auto select tool. So this gives you the ability to set a threshold value to threshold out automatically the different frames, and it will just run through every single frame in the experiment and keep the frames or remove them from the experiment. So again, very, very useful. This is kind of what a frame looks like, which is classed as a miss frame. So there is no diffraction on here, and you can see here that it gets the signal correct. There's no diffraction signal here. And then this is an example of a hit frame where we have particles which have got in front of the beam. That gives us our diffraction, and we have some recognized signal here. So this will be kept as one of our hit frames for our integration procedure. So once we add all those integrated images together, we sum all of those images, we get our composite diffraction pattern here. We then do our radial integration, and we get our 1D diffraction pattern. So just as an example, this is 16 frames from 632. And those 16 frames were 0.1 seconds. So this is only 1.6 seconds worth of diffraction data. And we're able to successfully index the salicylic acid powder data. If only just to give us polymorph identification, it's still very, very useful to be able to see which polymorphic forms are present in a mixed solution. And just to show you a real world example on aspirin, we've run the segmented crystallizer with aspirin as an example system. We chose aspirin because they have two very similar crystal structures. So their unit cells are very close. You can kind of see here the unit cell parameters are very close together. So their powder diffraction patterns are very close. And so they're quite difficult to separate in a powder diffraction experiment. So it was a good test of the diffractometer to see whether we could actually separate these two during a crystallization process. So we performed an anti-solvent crystallization from saturated ethanol with water as the anti-solvent. And we varied the solution anti-solvent ratio to tune the number of particles in each droplet. We then collected diffraction data continuously, segmented that using the frame select tool inside of Crystallis Pro, and then we integrated that data into our 1D diffraction patterns. And this is what the data looks like. So once you've integrated, if we overlay our CIF files for the two forms, we see that we get a nice separation of form 1 and form 2 around the 20-25 2-theta region. So we are able to say that we have a co-committant crystallization of the two forms, which is what we expect from an ethanol solution in mixed ethanol systems. So this data highlights that we can, even with quite closely matching crystal structures, we can still separate the diffraction heat profiles of two mixed phases in a mixed polymorph system, as I've shown for aspirin. So that was kind of the end of the examples I had. Just some conclusions, brief conclusions, and forward look. So I hope that I've been able to show our new facility dedicated to in-situ analysis of materials in flow, and that we're aiming to kind of bridge the gap between laboratory diffraction and beam time, and hoping to improve sort of the transition between the types of experiments that we'd like to do at Diamond, try to maximize the experimental time that we do at Diamond by allowing researchers to come to FlowXl and optimize their crystallization and data collection procedures before going to Diamond. I've showed two examples of the types of data we can collect. So the first example showed us that from the theophylline example, we can extract quantitative information about solution-mediated phase transformations for an API, looking at the dehydration of the monohydrate of theophylline. In the second example, I showed you this rapid polymorph identification in continuous crystallization systems. So this ability to do qualitative, not quantitative analysis, but to be able to fingerprint different forms or solid forms of the same material inside of a single droplet crystallizing in continuous flow. And I hope to spark some interest in the FlowXl facility and ideas for potential future experiments and collaborations. I hope it's been of interest. And just to end on, I'd just like to thank our sponsors, the EPSRC, UKRI, our collaborators, Nottingham and CMAC, who've done a lot of work with us to get some of the sample environments into the diffractometer, into the facility. The project group I've listed here, the people at Leeds are Fiona and Nick and myself and Dr. Alex Kulak. We're all based at Leeds. Chip Wilson and Alistair Florence were the people at CMAC. Nottingham was Karen Robertson, who's done a lot of work on the CRATE system, the continuous crystallizer. And the commission experiments, a lot of experiments done by people in the group at Leeds. In particular, Liam has done a lot of work on the segmented flow crystallization experiment I showed, and Hannah Bristow did a lot of the work on the theophylline. So thank you to you. And of course, Rigaku and Hariba who've done a lot of work on the actual development of the two systems as well, to be able to optimize them for our in situ crystallization processes. I've left my contact details here if people want to get in touch about anything in terms of in situ diffraction and using FlowXl in terms of collaborations. And thank you very much for your attention. And hopefully I've got some questions to answer. I'll be more than happy to take questions